Do you want me to shorten my talk, or what would you
No,
like me to do?
I just didn't hear the whole talk, and we can drive into the break time.
Okay, very good.
Thank you.
Well, yeah, in this talk, we're going to be stepping away from really from beekeeping and turning instead to how honeybees live in the wild, in nature. And you might be wondering, well, why do this? Why look at how honeybees live in the wild, all on their own? And the answer, I think, was well expressed for us by a gentleman named Wendell Berry. He wrote a book called Home Economics, and he wrote the following, and it really applies to any aspect of agriculture. We cannot know what we were doing until we know what nature would be doing if we were doing nothing. And I think he's right that we do a lot of manipulations with the plants and animals that we depend upon for our food and so forth. And sometimes we don't fully, at least I didn't, as I started out as a beekeeper, we don't fully realize how we're modifying the lives of our bees. And we've just seen a talk with commercial beekeeping, and there's a lot in there that we'll see is there's a lot of doing in beekeeping. And I think for us to understand what we're doing and fully understanding the impacts on the bees, it's good to see how the bees would be living if we were not doing anything and they were living all on their own. So that's what this talk is about. It's giving us going back to the natural history of honeybees and creating a solid foundation of knowledge of how they live when they control their lives. And as I say, looking at the natural lives helps us know what we are doing to our bees when we keep them in our hives. And I'm a beekeeper. I've been a beekeeper for 40 years, and I usually produce not tons of honey, but about 1,000 pounds a year. Here's a roadmap for my talk. First, we're going to review what I and others have learned over the past 40 years about the natural lives of honeybees. And then we will address the puzzle of how colonies are surviving in the wild. And we'll see that it's a combination of the bees having good genes and a good lifestyle, one that helps them survive and reproduce. Let me tell you a little bit about my study location. We're going to shift from the plains of Canada to New York State, over here in the northeastern United States. Now I'm showing you a map of New York State, probably to help you understand that when people think of New York State, they think of New York City often. And I want to explain that I don't live near New York City. It's not an urban environment where I live. It's up in the countryside of New York. It's a 5-hour drive by highway from New York City up to Ithaca. And the other thing that the reason I'm showing this map is the color coding of the map is useful for this talk because green means forest in New York and Pennsylvania and the other states of New England, like Vermont and New Hampshire and so forth. So where I live and work, there are vast forest lands. And the reason for that is that the soil, it's very hilly. We're getting into the Appalachian plateau region here. It's not good soil. The vegetation is mainly deciduous forest. The land is about 80% covered with forest, and the climate is one of hot summers and cold winters. It's a picture of my house, and it looks like this right now. Lots of snow. I've mentioned that the part of New York State that I'll be talking about is this Finger Lakes, or central region of New York State. These long lakes are, there used to be rivers, and they were gouged out by the last glacier to make deep lakes. Ithaca's at the foot of one of these lakes, Cayuga Lake, and you can see the heavy forest areas into the area south of Ithaca. It's good farmland up here. The glacier smoothed it out nicely. Down here, it's very hilly. And the reason I'm showing this map is primarily to identify, to refer to this Arnott Forest. This is a research forest owned by Cornell, and it's where I've done a lot of my studies of colonies, honeybee colonies, living in the wild, out in the forests. Here's a look at what this forested landscape looks like from the top of one of the hills. As far as the eye can see, looking south into Pennsylvania, it's just forest lands. it's great land for growing hardwood trees, sugar maples, oaks, and so forth. And that's the main industry up here is apart from people work like me, work at Cornell, is the timber industry. This Arnott Forest, as I mentioned, is a teaching and research forest owned by Cornell. So it's a kind of a preserve, and it lets me do studies here without having to worry about trespassing or anything like that. And my studies in this particular forest began, August 1978. What's that? That's 22 plus 21. That's more than 40 years ago. And my initial study was addressed at one simple basic question. How common are wild colonies? I'd been keeping bees since I was a teenager, and I knew there were wild colonies because I'd come across bee trees here and there. And I was curious about just, well, how common are these wild colonies? So I went to this forest and I located, mapped out in half of the forest, the wild colonies living in these big, tall hardwood trees. You might wonder, well, how do I locate wild colonies? I do it with an old hunting skill called bee hunting or bee lining. It's pretty straightforward, but it takes skill. You go to a patch of flowers somewhere, capture bees in a little contraption called a bee box, which enables you to introduce a bunch of bees efficiently to a square of comb filled with sugar syrup. The bees like that unless there's a honey flow on. They like that. The bees that you introduce to it go home. They'll advertise it with their waggle dances. And if the conditions are right, good weather, not a honey flow, you'll soon have a pile of bees on your comb. And then you can see which direction they're flying home. And you can work your way down the bee line step by step to their home. So step one, you're sitting there with your little feeder, you see the line, their flight line home, and then you follow it. Here's an example of a bee hunt that I did, not in the Arnott Forest, but it was just a demonstration. I did it on the Cornell campus. I started on a flower garden here, caught bees, honeybees off the flowers here, introduced them to my comb. I could see they were flying up to the north. There's a big tall building here, so I moved my feeding station with the bees over to here, and then it was really clear. They were going basically due north. And then every hour I moved the feeder close with the bees closer and closer and closer. And by the time I got here, it was only taking the bees a couple of minutes to leave my feeder, go home, offload the sugar syrup, and then come back. So I knew that bee tree was nearby. So then I just searched down the bee line, all the big trees, and I noticed bees flying in and out of a knothole up here. You might wonder, how on earth do you see bees flying out of a knothole that's that high up? It's because the bees are, you don't see the entrance per se. You see the buzzing bees, the moving bees outside the nest. By the way, if you're interested in this process of bee hunting, there's a pretty good book written on the subject. Anyhow, back to the Arnott Forest. This is the outline of the forest. You can see it's very hilly. It's mostly forested. Wherever it's green, it's forest. And one thing this map doesn't show is it's not just this forest is not an island of wooded hillsides. It's surrounded by more hillsides. In fact, it's hundreds of square miles of forested land like this, with down in the valleys some old farms. And there was one up on top of here. But it's almost all forest. And what I did when I first did my mapping of the bee colonies, I went to the back entrance of the forest and I caught bees off flowers there and got two bee lines going. One went to the south and one went to the north. So there were two bee trees. And with about 2 days work, I could work my way down from that point down to the specific tree that the bee's home. So I found that one and I went back here, started over, but this time I followed the bee line north, found this tree. Then I shut down all my operation here. I moved about a half a mile down the road, started again with fresh bees off a fresh patch of flowers, found that some of the bees were going back to one of the trees I'd already found. Others were going up to this tree. So I found my way up to that, worked my way up to that tree. So I did that. And spent about a better part of 3 or 4 weeks working my way around this half of the forest. And you can see I found 8 bee trees. And just some examples of what I was finding. This is one I call the easy ash. It was easy because its nest entrance was easily seen from the ground. So it was easy to see where the bees were going, where their home was. Here's one that was a mystery maple. I call it the mystery maple because the bees were their nest entrance was high up in this tree, and I couldn't see it until I climbed up into this pine over here and looked across and could look down at it. And then I saw the bees were diving down into a crotch in the main portion of the tree where there was a crack in it. And here is one that I call the deceptive aspen because it was a small tree. I usually don't find bees living in trees that are only about a foot at diameter at breast height, but there they were. So it's a great fun to go bee hunting. It's a treasure hunt. Takes time, takes skill, but it's rewarding. And it turned out to be very useful to have this knowledge about the abundance of the wild colonies. Here again here is a typical bee tree home of a wild colony. It's in a big tree. It has a high entrance off the ground, there's small entrance, modest nest cavity, 10 gallons, the volume about one deep Langstroth hive body. And it's got thick walls. Usually they're in old trees with thick walls. We'll come back to that, all of these traits. So just to emphasize, got about 7 square miles. I found 2.5, searching half the forest, found 9 colonies. That works out to 2.5 colonies per square mile. So that was a baseline for 1978. And the reason that that's important is the Arnott Forest is the only place that I know of in all of North America where we had information about the abundance of wild colonies pre-Varroa. Pre-Varroa. In my part of the world, I first saw Varroa in 1994 on some of my bees. And so I assume it came in around 1993. And the first 2 winters following the appearance of Varroa in my colonies, I didn't know what to do. And I lost both winters. I lost more than 80% of my colony. So I figured that probably out in the wild, and then I could started treating the bees, and then things got better, treating them for Varroa. But I knew there was nobody treating bees out in the woods, out in the forests. So I thought, gosh, probably all those wild colonies have been killed off. That's really sad. But in 2002, I said, okay, Tom, don't just assume that. Let's go check it. So in 2002, I went back to the Arnott Forest and I repeated the bee hunting that I had done, 24 years before. And I found 8 wild colonies. I really expected to find none, but on the first day, I again found bees on the flowers up in the forest area and lined out 8 colonies, covering the same area. And I went back again in 2011 for to do a study to get more samples of the wild bees from the wild colonies. Again, searching half the forest, and I found 10 colonies. So it had become quite clear, even back in 2002, that this colony was somehow persisting, but it was unclear how they were doing it. And of course, one possibility is, well, maybe these colonies were not infested with the Varroa. And so back in 2003 and 2004, I checked to see if the colonies living in the Arnott Forest had Varroa. And the way I did this is I put bait hives up in the Arnott Forest to check for mites. Now I'm going to talk tomorrow about bait hives, and I'm not going to advocate the bait hive now that I was using back then. This is you can see it's a 1-story deep Langstroth hive. I've put it up in the tree. That's bees like that. Bees like their nests high. I've given it a small entrance, and I get good acceptance rate with a bait hive like that. But the key point here is I was putting them up because I wanted to catch wild swarms in the middle of the forest. And and I should have mentioned earlier on, the nearest managed colonies are about 5 miles away from the northern boundary of the Arnott Forest. So I put these bait hives deep in the Arnott Forest, thus many miles from the nearest managed colony. So I'm pretty sure the bees I've caught in the Arnott Forest are from the wild colony population. Put them out to check for mites. Here's what I found. In the summers of 2003 and 2004, I caught 11 colonies in my bait hives. And since the bees are on movable frames, I could inspect the combs and so forth, and the brood combs. Varroa was in 100% of the colonies. Chalk brood cropped up in 1 of the 11 colonies, and American foul brood and a European foul brood did not show up in any of these colonies. but the main conclusion was that the wild colonies do have varroa, and yet they're surviving, they're persisting up in this forest. What's going on? How is this population of wild colonies in the Arnott Forest persisting? Well, when you think about things biologically, of course, we all know that there's one part of a store of an explanation about anything biological is maybe at least the genetics of the story, but then there's also the environment or lifestyle factors of the story. So if we look at the good genes side of the possible answer to this question, we're asking the question, do bees have genetically endowed resistance to the mites? And if we look at the lifestyle, we're really looking at the question, how do these bees live? Are there ways that they're living that help them deal with the mites? And we're going to go through colony spacing in the wild, nest site, nest structure, and other factors that help the colony, that are helping the colonies survive despite having mites, varroa mites. But we'll start with the good genes. Or do they have genetic mechanisms of resistance? So, key question next. Have the wild honeybee colonies experienced strong natural selection for varroa resistance? And as we all know, what natural selection does is it sorts among genetic variants. And the units, in this case honeybee colonies that have good genetics to deal with varroa, will survive and reproduce. Those without the right genes will not survive and reproduce. And so over time, the colonies, in principle, should develop mechanisms, genetically based mechanisms of resistance to varroa. And the answer to that question is yes, they have experienced strong selection. And the reason I know this, and it's remarkable that we have the evidence, is that back in 1977, when I was starting to get interested in how bees live in the wild, I went around, I was capturing swarms with my bait hives, and I was finding bee trees. And so I went, and every time I caught bees to set up a new colony from a wild colony, be it a swarm or whatever, I took a sample of the bees and of the workers, and I pinned them, and I put them in the Cornell University insect collection. So sitting for some 40 years in the Cornell insect collection were pinned specimens of honeybees that could be analyzed genetically and compared to modern specimens that I collected in 2011. So I had before and after samples of varroa, and using some very fancy, sophisticated techniques done by one of my students, who's now a professor at the Okinawa Institute of Science and Technology. He could look at the genetics of these bees, looking across the whole genome of each bee, fully across the whole entire genome, and compare the genetics of the pre-varroa bee and the post-varroa bees. And just to clarify where these bees are coming from, they were sampled in 1977 and in 2011. And in both cases, I had sampled bees from 32 wild colonies. Many of them were the wild colonies were down in the Arnott Forest. Others were in the hills and forests outside of Ithaca. What did we find? Well, one of the most striking things that we found, and this is based on the part of the bee's genetics which are in their mitochondria. So this is based on mitochondrial DNA, which is only passed on from mother to daughter. So we found that the number of queen lineages shrank dramatically between 1977 and 2011. That's shown in this diagram, which shows you a kind of pedigree or genealogy of the queens in the 31 colonies from 1977 or 2011. And the 1977 queens had a diverse set of lineages. They came from different queen lines, mother lines. But in 2011, there were only really 3 distinct queen lines populating in the population. There was one colony was represented by this queen line, and all the rest of the colonies were represented by this queen line and this bunch of this queen line down here. And this is prima facie evidence that this population of wild colonies experienced a population bottleneck between 1977 and 2011. And it's all, and that's almost certainly was due to the arrival of Varroa, because we were all watching colonies die off. And but the second part of the story is what I'll turn to next, and that is that not only did this population of colonies experience a bottleneck, but it also experienced strong natural selection. How do I know that? I know that because Sasha is able to look across every chromosome of these bees, both the historic 1977 bees and the 2011 bees, and these are readouts for each of the 16 chromosomes in honeybees. And wherever you see a red dot, that means there is a gene that changed so much between 1977 and 2011. It couldn't have happened. It could not, that change could not be just by chance alone. It's a marker. Extreme change is a marker of natural selection. So there are 634 sites on these chromosomes that show strong signatures of natural selection. We don't know in detail what any of those genes do, but we do know in general that half of the genes are involved in the development of a bee, so they determine the traits of the adult. But we do know something, so we don't have a precise picture of how the genes have changed, just we know lots of changes have occurred, but we can get some indications of what these changes have been. There have been morphological changes, for example. Here we're comparing the widths of the heads of the modern 2011 bees versus the old 1977 bees. The bees are a little smaller now than they were before. Head widths are smaller. Also it's called the intertegular span. It's the span between the wing joints on the thorax. That span is also a little smaller. Another marker of how the bees are a bit smaller now post-Varroa than pre-Varroa. There are also behavioral changes between the old bees and the new bees. And this doesn't come from the genetic analysis, this comes from our behavioral experiments. And what we know is that, and I'll be talking about this more later in the talk, that the wild colonies workers are good at killing adult mites, and also they're good at uncapping infested brood cells. They bite the legs off the mites, and they have a little nice trick where they uncap infested brood cells, leave it open for a little while, and then go back and recap it. And why that disrupts the mites' reproduction is not clear, but it does not, but it does, and it does it without disrupting the development of the bee inside the cell. It's pretty slick. Now, what I want to do now is share with you the evidence that I have that these colonies that are out there in the wild do really well. I'm going to give you real numbers on this. So what I'm going to share with you now is a comparison of mite counts and colony survival for colonies that I don't treat and that are headed by a queen from one of three sources. So I'm going to have going to set up an experiment where I have three groups of colonies. The groups differ by the source of their queen. In one case, the queens, and I'll call these the wild stock colonies, their queens came from bait hives that I placed in forests outside of Ithaca, New York. So I don't know exactly where these queens came from. I just know they were caught in a remote part of the woods south of Ithaca. The second group of queens are queens that I purchased from a treatment-free queen producer in Vermont. His name's Kirk Webster. And he uses a Russian stock, and he hasn't treated his bees for 20 years, but he makes packages and sells honey. So his bees don't need treatment. The third group of queens were a set of queens that I purchased from a large commercial queen producer in California, Oliveras. And these were the Italian stock they sell. And this company does treat its colonies with miticides. So we've got two groups of untreated queens from untreated stocks and one group of queens from a treated stock. And when I did this experiment, I had three groups of colonies and I put them all in the same environment. There was three, you call them mini apiaries, all put along this hedgerow with about 100 meters between each group. And each colony, so there's a shared environment, basically. Each colony was started as a two-frame nuke in June 2019, and the bees and brood that I used to start each nuke, they came from all came from the same shared colonies, same source colonies. They were not, and these were the source colonies were just shared, and I don't know if they were resistant or not, but the test really is based on what the workers do that come out of these queen's eggs. And each colony was left alone all summer. So they were set up in June 2019 as each one is a two-frame nuke, 2 frames of bees and brood and food. Each colony was left alone all summer with its queen. And I want to stress this, there are no treatments given for varroa to these colonies. Now here are the results. And we're first going to look at the results at the end of this summer in October 2019. Here again, so we've got 3 types of colonies: wild-caught queens, Kirk-Webster queens, Olivares queens. And here's the sample size. Small samples, but it's still telling. Number of colonies that were queen-right and ready to go into winter in October 2019, 657. And here we start to get some evidence or results. These are the mite counts, mites per 300 bees, measured in October 2019. I use Randy Oliver's method of doing the wash using the soapy water with Dawn. And you can see, okay, so we got some variation here. We got 3, 22, 5, 1, 1, 1 for the wild-caught colonies. Kirk-Webster's colonies, the colonies with Kirk-Webster's queens, 1, 3, 0, 3, 1, pretty low. The Olivares VSH queens, 18, 15, 9, 24, 4, 10, 17. So rather high, rather high. And now what do we see next? We're going to look at the percent colonies alive in April, the following spring. And in this diagram, red means dead. So of the wild-caught, the colonies headed by wild-caught queens, 1 was dead, and it was the one that had a very high mite count the last October. The rest were all alive. All of the Kirk-Webster colonies were alive in the following spring, and there was only 1 of the Olivares, of the colonies headed by the Olivares queens, that was still alive. And that colony knew how to deal with the mites very well. And so it's not surprising that that one did survive. What is telling, though, is that the difference between the Olivares queen versus the Webster from a non-treatment queen producer, and also compared to the wild-caught colonies. So what I conclude from this little experiment is there was greater heterogeneity among colonies headed by wild-caught queens than commercially produced ones. But there were lower mite counts in the colonies with the wild-caught and Webster or queens versus the ones with the Olivares VSH queens. And again, these first 2 categories these are queens from colonies or lineages that have not been treated for varroa, and this is from a lineage that has been treated for varroa. And we see very different levels of winter survival, much higher for the wild-caught and Webster queens than the Olivares queens. I don't know. So those are pretty striking results, and I'm not surprised. It's what I would expected, but it was good to get numbers on this. So back to this little table, how to address this question: how are these wild colonies able to persist without being treated for varroa? Well, as I said, part of the story could be that the bees have good genes, that they're resistant, that gives them resistance to the mites. And the answer is, to that part of the story, the answer is yes, they do have good genes. That's what's shown by that last experiment. Let's turn now to the aspects of lifestyle, colony spacing, nest site, and nest structure. I'm going to tell you about an experiment which involved comparing the situation of colonies that are dispersed in a forest, like we have in the Arnott Forest, where I didn't stress this, but these colonies, they're about 1,000 meters apart, so they're more than a half a mile apart on average. Sometimes they're closer, sometimes they're farther, but they're pretty widely spaced. In contrast, here's one of my apiaries, and this is a big change. Here they're like a few meters apart at most. And of course, so As we all probably appreciate, apiaries are a major change in the ecology of honeybee colonies, and crowding them together like this certainly helps it spread parasites and pathogens. But I really wanted to know just how powerful an effect that is to have colonies crowded in apiaries in terms of disease spread, dealing with Varroa in particular. So I did an experimental study, which I'll share with you now. It was to study the effects of colony spacing of drifting in bees on spread of disease. And the questions are simple. If dispersed, is there less drift of bees among colonies? And if dispersed, is there less spread of diseases, e.g. Varroa, through drifting? Here's the experimental setup. I set up two groups of 12 colonies, and again, I start out my colonies as a 2 or 3 frame nuke. All of the queens came from the same queen producer in California. So I had 24 colonies with similar genetics, queens from the shared, at least from a shared source. In one group, I set up my colonies in an apiary. In the other group, I set the hives more widely spaced, much more widely spaced than in the apiary. And here's a bird's eye view of the experimental setup. The apiary is here. So you can see that image is represented by this little bar here. And then the other colonies, the dispersed colonies, are dispersed out across this long, long field. This is an old radio antenna array that Cornell had set up, and I took it over and put all these little places for my colonies. So some were dispersed, some were crowded. Two groups of 12 colonies. I set these colonies up in the spring of one year, and I withheld Varroa treatments from these colonies during the 2 years of this experiment. I really wanted to see what the effect of dispersal on dealing with a difficult situation of having Varroa. And first thing I did is I looked for a difference in drone drifting. And the way I did this is when I set up my 12 colonies in each group, I set up the colonies such that 10 had a Wooten golden Italian queen. And I don't know if you know about those, but they're queens that they're homozygous for a mutation called cordovan. So the drones, all the drones they make, which are hemizygous, they're all cordovan. They're bright yellow, and they really shine. They're quite bright. But the other two colonies had New World carniolan queens, so all of their drones are dark brown. So I could see, so two of these colonies, and in both groups, it was two colonies in the center of the array. This one and this one produced dark drones. Likewise, there were two colonies in the center of this array that produced dark drones. And so I could look for drone drift just by going to the other colonies around either in the apiary or in this setup to see whether I saw any dark drones flying into the colonies that were making only yellow drones. And what I found was there was a big difference in the level of drone drifting. 24% of the drones in the colonies that were making only yellow drones were dark. This 24% is based on counts of drones flying out of the hives. Whereas in this setup over here, nearby, but where the colonies are dispersed, 0% of the drones in colonies with golden Italian queens were dark. So there was no drifting, no detectable drifting when you just put the colonies, what do we got here, about 30 to 50 meters apart. So big difference in level of drone drifting. And what does that mean for disease? Well, it means also that there's going to be a difference in, almost certainly a big difference in the level of worker drifting, and thus the spreading of mites if one colony picks up a high level of mites. So much drift on the one hand, and little, if any, drift in the other situation. And as I say, I expect the same difference with respect to worker drift, much versus little, though in this experiment I couldn't measure that. But what you're probably thinking is, okay, well, that's interesting, but what's the effect on the colonies and particularly their survival? Was there better colony survival in this situation if the colonies were spread out? Well, as I said, I didn't treat these colonies during the duration of the experiment, and that was for 2 years. And at the end of 2 years, in the crowded group, 0 out of 12 were alive after 2 years. And the dispersed colonies, it was different. 5 out of 12 were still alive after 2 years. The queens came from the same source, so I don't think this is a genetic difference. I think this difference in result um, arose from the uh, less drifting of bees between colonies and probably perhaps less robbing when the colonies are more dispersed. So the conclusion I've drawn is that spacing colonies more widely helps their survival, probably by reducing bee drifting and mite spreading. So back to the broad question, how are the wild colonies able to persist without being treated for varroa? We can add to, on the good lifestyle part of the story, it's clear that colony spacing plays a role as well. It's part of how these wild colonies are surviving. They're living widely spaced. What about the nest site? Well, here's a couple of nest sites of these of a couple wild colonies. Seen this picture on the right once already. One feature of the nest site of wild colonies is the size of their nest cavities, which they're relatively small. They're about, as I mentioned before, about 1 deep hive body, but I'll describe it more precisely here. This is a distribution of the volumes of the nest cavities of 21 wild colonies that I collected and dissected. I collected the nests, split open the tree cavities, the trees, took out the bees and the combs, refilled the cavities with sand, and so I could measure very precisely the volume of the cavities. And this is the distribution. Most of them are between a little bit smaller or a little bit larger than 1 deep Langstroth hive body. There was one exception, which is a kind of a puzzle to me, but none were as large as 3 deeps and certainly not 2 deeps either. So the natural size of the nest cavities in nature is small. And by the way, where does that distribution come from? Is that just by chance? Maybe there aren't very many really large cavities or really small cavities. No, it comes about because the bees have a preferred cavity volume. If you give them a choice, and this was part of my work for as a graduate student, I studied the nest site preferences of honeybees. And the way I did it is I put out boxes that were identical in all ways, sets of boxes in which the boxes in the set were identical in all ways, except in this case, the volume. So the color was the same, the shape was the same, the entrance size was the same, but just the cavity volume was different. And I put these boxes out, and I didn't put them out this close together. I actually put them along hedgerows about 20 to 30 yards apart. And when I did that, and this choice was based on the fact that I saw very few 10-liter nest cavities in nature and very few large ones. And what I found was that yes, this is what they preferred. They never moved, no swarms moved into the 10-liter box. One swarm moved into all my 100-liter boxes, and most of the move-ins, which was about 10, moved into the 40-liter. So it was a very clear preference by the bees to live. When they get a choice, they want to live in a nest cavity that's about that size. Now, okay, so we know that they live in nature in smallish nest cavities. And as beekeepers, we all know that any colony left in a single deep hive body is going to swarm. And we just heard a talk where much of the management protocol reflects that need to expand the nest, expand the hives, give the colonies lots of growing room. But in nature, nobody's putting honey supers on the bee trees, so we expect that there's frequent swarming. We can be virtually certain that these wild colonies are swarming frequently. And I have data on that. And yes, indeed, they swarm about 93% of them swarm every summer and cast after swarms. So, but the question becomes really, does this frequent swarming lead to fewer varroa? And the reason one might think that is because when a colony swarms, there is a broodless period in there. When all the brood has emerged, but before the new queen has started her laying. And certainly there's a period in which the colony has no sealed brood. And swarming also exports a lot of mites. We know that when the prime swarm leaves a colony, it's about 70% of the worker bees leave. And a lot of the mites are on the adult bees. And so that's a big exporting of mites from a colony when it swarms. But to really look at this effect of colony size on mites, I again had to do an experiment. And it's again, it involved two groups of 12 colonies. And again, the queens from all these colonies came from the same queen source, commercial queen producer in California. 12 of the colonies lived in a 1D hive body. The other 12 colonies lived during the summer in 4D hive bodies and in the winter in 2D hive bodies. So these were my honey. These colonies produced a lot of honey for me. These colonies produced no honey for me. And all 24 were established simultaneously in 2020. All 24 queens, one queen producer. And I just let these colonies go. They were set up in, I think it was June of 2012, again, with just as two frames of bees and brood and a new queen. And I can't go through it in detail, but I'll just tell you what happened the summer of 2012. None of the colonies swarmed because they were just building up all summer and storing up food. Summer of 2013, the colonies were strong, and 10 out of 12 of the colonies in the small hives did swarm, and only 2 out of the 12 in the large hives swarmed. So 83% versus 17%. So no surprise, living in a smaller cavity leads to more swarming. Everybody knows that. But what was less clear was what was the effect of this difference in hive size and on the mite counts in the colonies. And here's the mite counts. Here are the tracks of the mite counts in the colonies. And again, it's mites per, in this case, I just simplified it to mites per 100 bees, so we can do a percentage. We can see that when things started out, both colonies had the same low level of mite counts for their mite counts. And it was low the following summer, following spring, but then things diverged in the summer of 2013 because now the colonies were well established and the mites were well established. And in the large hive colonies, the mites went up and up and up. And by August, they were getting dangerously high, and they got even higher. And the only reason it didn't go higher is many of the colonies were collapsing. Whereas in the small hive colonies, the mite counts never went up very high, and they were going down in August. And so at the end of the summer, 6 mites versus 1 mite per 300 bees. And what about colony survival over the winter that followed? In the large hive group, only 2 of the 12 colonies survived, and there was one colony in there that was an excellent varroa killer, by the way. And in small hive group, it was 10 of the 12 colonies that survived. I'll just add parenthetically that one colony that was an excellent varroa killer was also an extremely defensive colony. So they're good at defending themselves from varroa and from beekeepers. So coming back to this table, how are the wild colonies able to persist without being treated? Part of the story is good gene, part of the colony spacing, and part of it is nest site or size. And there's one last topic we'll consider, and this is the nest structure. Here we are, here's our nest, entrance high off the ground, looking inside this nest cavity, relatively small, thick-walled home site. Well, what's the effect of these thick nest cavity walls? And in nature, they can be quite thick, typically 4 to 12 inches thick, somewhere in that range. And I have just tracked cavities that have in a box. I have two cavities which I tracked, put a thermometer in them, electronic thermometer to track the temperatures. And in one of the cavities was a cavity that I cut out into a large sugar maple tree into a rectangular parallel piped shape, a regular square-walled shape of 40 liters. And its temperature recording in April of 20, and that's our spring, of course, was very stable in that tree cavity with the thick walls. What these other tracks are, the black is ambient. So outside the cavity, it's jumping all around. It goes up and down every day. And interestingly, in the box, and I had a box outside that was built out of just 1-inch planed lumber. So it's just the same material as our hive bodies are built out of. and it jumps around as much. There are no bees in any of these cavities, but it just goes to show that the cavities we put the bees in have very little buffering of the swings in the ambient temperature. And there is I don't have time to go through it, but when we put with this better nest insulation in the tree cavity, the temperature is more stable, and in the winter there's less condensation on the bees. Let me show you, to talk about, just mention briefly this difference in condensation. These two images show you, give you an introduction to the effects of insulation on condensation in a hive in winter. This is a gentleman's beekeeper, friend of mine, and he did a simple study where he set up his hives, and on top of each, instead of an wooden inner cover, he put a sheet of plexiglass atop the second super of his overwintering colonies. And in one of the hives, he put nothing except the outer cover over his inner cover, which was this sheet of plexiglass. In the other hive, he laid a sheet of 2-inch foam over this inner cover, and then of course covered that with the outer cover. And I think you can see that the difference in between these two is striking. There's frozen ice up here, and there's water dripping down in the cluster area here, whereas up here the bees are clustered up tight against that insulated surface up in the top of their hive. And so this is something we're just starting to learn more about, is really the impact of good insulation in terms of on the colonies. He incidentally records the temperatures inside with these devices called hive minders. Another point of comparison inside the nest is the cell size. A lot of people ask me about this. Are the colonies in the wild, do they have small cell combs? Well, they're slightly small, but they're not. They're 5.19 millimeter wall-to-wall dimension on average, whereas our commercial foundation gives us 5.4 millimeter wall-to-wall. So they're a little smaller, but it's nothing like the small cell comb, which is usually considered like 4.8 millimeters. So the combs in the wild, the brood combs in the wild, cells are a little smaller, but not hugely so. Another thing that's different is in the wild, there's propolis coating the walls and the ceiling and the floor. And to show you what the propolis is covering up, I've chipped away the propolis in this portion from this wall of a bee tree home. You can see it's fibrous, rotting. Well, not rotting because it's quite dry, but it's got a lot of places. If it gets wet, that microbes and yeasts could grow, whereas the propolis seals all that off. Sometimes the bees will even go to the extent of coating with propolis the floor of one of these bee tree cavities. And you can see there's been a squirrel nesting in this cavity before the bees moved in. Lots of his or her shells. So what are the consequences of this natural nest structure? Well, I think the consequences of living in the small nest cavities with good insulation and much propolis are as follows. They have small brood nests and small colonies. Do they have more colony reproduction through swarming and drone rearing? Yes, they do. Do they have less thermal stress? Evidently so. Their nests are buffered against the ambient swings in the temperature, and they have good insulation, better insulation. Is there less disease stress? Almost certainly so, particularly for stress diseases like European foul brood and chalk brood, where if a colony gets chilled, those diseases can take off, as I'm sure you all know. So to come back to this whole panel here, remember the basic question is, how are these wild colonies able to persist without anybody treating them for varroa? They're all on their own. And the answers were, generally speaking, it could be good genes and or good lifestyle. And actually, it turns out that these bees have a lot going for them when they live in the wild. They've got resistance through natural selection. They live widely spaced, which helps them. There's less drifting, less mixing, less spreading of disease. The nest site has the features of better insulation, entrance, and a small cavity, which promotes swarming, which helps. And then there's just the whole nest structure itself, including the walls of the nest. So to summarize what I've presented, I think I can do it all in this one page. First point, colonies in the wild can survive without chemical treatments for varroa. And I've given you the example of the Arnott forest bees, but at least where I live, back here in the eastern US, where there's lots of deciduous forest, in fact, most of the land cover is deciduous forest. This is true wherever people have looked. There is a population of colonies living in Pennsylvania, eastern Pennsylvania, where the colonies are there living in the wild just fine. One of my neighbors, who's an excellent bee hunter too, um, went into one of the state forests that contains old growth forest, and he mapped out the bee colonies in that one, and he found everyone living in a hemlock tree, high up in the hemlock tree. He's a very good bee hunter. He's also a very good tree climber. So colonies in the wild can survive without chemical treatments for varroa, and the reason they can do this is because we now see that they've experienced strong natural selection in the last 20 years, probably almost certainly due to varroa. Remember, the genetics tell us that these populations of wild colonies, at least the one in the Arnott Forest, went through a population bottleneck, which is a signature of powerful selection, natural selection. And the third point of summary is that the colonies live in nature differently than they do in our hives, and this is helping them too, we now know. On the one hand, they are not treated for varroa, and the benefit of that in the long term is it allows selection for resistance. They also live widely dispersed, which has the immediate benefit of less spread of disease from when colonies collapse or come down killed by American foul brood. They occupy small nest cavities, which means they have smaller honey stores, but they also have more swarming and better colony health. And that swarming, that brood break, that exporting of the mites is as far as I can tell, that is critical to this whole survival of these colonies in the wild. But they also benefit from having things like thick nest cavity walls for better insulation of the nest and the thorough coating of the nest walls with propolis. And this provides the bees with a stronger natural defense against microbial infections. And there's work done by Marla Spivak's laboratory at the University of Minnesota that shows that in an experimental setup, when the bees have rough walls on the insides of their hives and they put lots of propolis down, the genes that are in those bees, in those colonies, the genes in the bees and colonies that have their walls covered with propolis are working less hard to build defenses. Their disease resistance traits are less strongly expressed in those colonies. So they can have more resources to devote to other things. For more information on this topic of how bees live in the wild, I refer you to this book. It came out in 2019, and I'm kind of proud of this book because I think it's the only book that has ever been written about how honeybees live in the wild rather than under a beekeeper's management. And I'm not putting down beekeeping because I am a beekeeper, but I think it's good for us, as I said at the beginning, I think it's good for us to have a perspective on what we're doing as beekeepers. And the best way to get that perspective is to understand what we're undoing. And I'm learning, even though that there are ways to undo some of our beekeeping methods in ways that, yes, it will reduce their productivity, the production of honey by colonies, but it will give them better health. So it's like a lot of things. There's a honey health trade-off in the way we manage our bees. Okay, I think I've used up my time, so I will end here. And if you'd like to do questions, we'll give that a shot. Okay. So let's see, Q&A, I'll click on this. Dr. Seeley, did you determine the parentage of the wild-caught honeybees in your varroa study? Is it possible they were also of Russian stock, or were they Italian line? Let me think. No, we did not do a genetic analysis of the parentage of the wild-caught bees that I used in my varroa study. No, that would have been good to do, but I didn't do that. Next one. Did you do anything to mitigate any potential crossover between hives through common equipment used to check the hives? Um, yes, I would clean the hive tool. That was the only equipment I used was a hive tool. So I learned that a long time ago from the New York State bee inspector. Take a bucket of water and soap and water with you and clean your hive tool between hives. Third question. Does there seem to be a cost to selection of genetically resistant strains? i.e., are these strains less productive honey producers, more defensive, more prone to swarming? I don't know. That's a very important question, and almost always, um, when you get something that's good, it comes at a cost, and I that's a really good question to address. I do know that my hunch is they're no less productive of honey. I mean, they're good at making honey because if they're not good honey producers, they will die. So there's been good selection for them to be good at making honey. They may be more defensive, and that's certainly what we saw in the variation in some of our experiments where we didn't treat colonies, and we found that the colonies that were killing the mites really well were very more defensive than the ones that were succumbing to the mites. So more prone to swarming, that is also a possibility, but I don't know the answer to that. Very important question. Great. Looks like we've worked through the questions. So I will stop share and turn it back to the hosts. Oh, here's another question. 20 years is a relatively short period to see genetic effects of natural selection. Do you believe we'll see the genetics of Africanized bees take over? Um, two-part answer to that question. The 20 years is actually, it is relatively short compared to some things, but when you think about the strength of the natural selection that Varroa has imposed on the wild colonies, where 80, maybe 90% were killed off, it's pretty clear that that's a very powerful selection, so it doesn't take a lot of time. With respect to whether Africanized bees take over or not, that will depend upon where you are. Where I live, the Africanized bees don't live, they don't thrive. Every once in a while, a migratory beekeeper will bring some bees up, but those bees die out. They don't take it in the winters. But in some parts of the United States, they will take over, and they certainly have taken over, such as in Arizona. Becky Mack, I was told that in California, they raise the queens constantly and encourage beekeepers to change out the queen spring and fall. Is there a chance that changing out the queen so often causes weakening of resistance? I doubt it, but I don't know. Can't be sure. It might, if it creates a brood break, it actually could contribute to resistance to mites. But I can't give you a firm answer. I'm sorry. Okay.